and for that echo. And thank you, all of you, for inviting me and having me here today. So I'm going to talk to you about the evolution of complex multicellularity, which is this transition from single cells to differentiated groups of cells. For two of these key steps that I'll be focusing on are group formation and cellular specialization. Now, during this transition, selection is changing from acting on the level of the single cell to acting on the level of the multicellular group. Now, multicellularity has evolved dozens of times across the tree of life, including dozens of times within the green algae alone. So why is this transition so common? And do similar processes underpin repeated instances of multicellular evolution? And the processes I'm specifically looking at are interactions between genes, development, and the environment, and how they shape the repeated evolution of multicellularity, examining both group formation and cellular differentiation. So I use the Volvocene algae as a model system. These are an awesome model system for studying the evolution of multicellularity, as Will said. You've got species like Chlamydomonas that are unicellular most of the time, but can also form groups in response to the environment. You have species like Gonium here that are multicellular most of the time, but are sometimes unicellular in some environmental conditions. There's species like Euterina that are undifferentiated. So each one of these cells here is capable of dividing and giving rise to the next generation through asexual reproduction. All of these cells are undifferentiated. They're similar to a Chlamydomonas cell. But in some environmental conditions, they will differentiate to be small and somatic-like. Some of those cells will. Similar to the small cells of this genus Cladorina, or the small cells of these species of Volvox, which have full germ-soma division of labor, specialized germ and somatic cells. So we can study the evolution of group formation in this clade really nicely because group formation, obligate group formation, has evolved at least twice. You don't need to worry about the species names here, but everything in general corresponds to a species that is characterized as being obligately or at least predominantly multicellular. And everything in yellow is unicellular. So you can see there's a transition to multicellularity here and here. This is work that was done by Ross. And we know that group formation in the unicellular green algae is likely or this group formation response to the environment is likely ancestral to Volvocene green algae. There's a wide array of unicellular green algae species that form groups in response to different environmental conditions, such as predation. And they can form these groups in different ways. So Chlamydomonas will form these large aggregative clusters like you see here, in response to the presence, this is not the right PowerPoint. 1 second here. They'll form those large groups in response to the presence of predators. And this is the correct PowerPoint. Sorry about that. And you can see the dynamics of this group formation here. So when you add predators, you'll see groups form within about 10 minutes. These are growing in a day-night cycle. In the dark, they break apart. I don't really understand why. It's a big mystery. And then they come back together. So this is group formation through aggregation. They also do group formation through clonal development. So they'll form these non-motile groups that look very similar to the obligately differentiated multicellular species in response to stressors such as salt. So what are the molecular mechanisms that are underlying cell group formation in response to predation? And could those molecular mechanisms that are involved in aggregation be co-opted for obligate clonal multicellularity? So we conducted RNA-seq on triplicate flasks of Chlamydomonas that we cultured with their predators, which are these little rotifers called Daphnia. And we also did RNA-seq on the controls. And we found that 131 genes were significantly differentially expressed. This corresponds to about 0.78% of the Chlamydomonas genome. And we focused on one candidate gene that we're calling CGR1. This is a transmembrane protein. It's a potential cell adhesion factor. And its expression increases after the addition of predators, which you see here. It also has a DNDS of greater than 1, which means that it's been subjected to positive selection in the Volvocene clade. So we ordered CGR1 mutants from something called the Chlamydomonas library. We ordered 2 of them, and we cultured them with predators to see if they could aggregate, like the wild type does. And we found that they had a reduced ability to aggregate, indicating that this gene is necessary for normal aggregation. Then we took the gonium version of this gene and its native promoter. We transformed it into Chlamydomonas, and we found that Chlamydomonas with the gonium, CGR1 and promoter, formed these large disorganized groups that are consistent with the aggregative structures that we see in Chlamydomonas in response to predation. We also looked at whether this gene was expressed in multicellular Chlamydomonas that Will's lab had evolved through settling selection to have a multicellular component of its life cycle. And we found that CGR1 was one of the genes that they identified as being significantly differentially regulated in the multicellular Chlamydomonas. And you can see here that its expression is increasing substantially in the period of time in which the colony is growing larger. And here's a picture of what these large multicellular colonies look like. This is very similar to the aggregative Chlamydomonas. Finally, we examined the expression of CGR1 in multiple multicellular species, which is gonium and pandorina here, and compare that to the wild type expression in Chlamydomonas. And we see that there's changes in the regulation of this gene and when it's expressed. In Chlamydomonas, it's expressed during mitosis. In gonium and pandorina, it's expressed throughout their life cycle. So there's changes in when this gene is being expressed. So what we think might be happening is that simple multicellularity in the Volvocene algae might evolve through the modification of aggregation pathways. And specifically, I think what might be happening is that you've got these mechanisms involved in aggregation in response to the environment that are being incorporated into an existing clonal development pathway. So you've got this reorganization of multiple developmental pathways that could be leading to the evolution of obligate clonal multicellularity. So I told you I was going to talk about 2 different stages in the evolution of multicellularity: group formation and cellular differentiation. So cellular differentiation is environmentally regulated in unicellular organisms. They can change their state in response to the environment. And it's developmentally regulated in multicellular organisms. But what mediates this transition from the environmental regulation of cellular state to the developmental regulation of cellular state? Well, one possibility is plasticity. That is, cell-level changes in cellular state and cellular morphology in response to environmental cues. So a lot of my research focuses on whether the modification of plastic responses to the environment shaped the evolution of differentiated multicellularity. And more specifically, I'm interested in whether new cell types can arise as a plastic response to the environment, which you should see here. Then that plasticity might be lost or stabilized through a process called genetic accommodation, which just means changes in plasticity. Then it could be secondarily refined by adaptive evolution. So you can see changes in traits like cell number, for example. And I'm also interested in whether obligate somatic differentiation in the Bulbocene algae evolved through the modification of stress response pathways. So cellular differentiation has been really well characterized in the Bulbocene algae. There have been multiple gains and or losses of somatic cells. You take a look at this phylogeny, you don't need to see any of the species names. Just know that everything in teal is undifferentiated. Everything in yellow or purple has somatic cells. And I really want to draw your attention to this smaller subclade here. This is the EVP clade, which stands for Uterina, Pleidorina, and Volvox. So this clade, as the name suggests, consists of 3 genera, Uterina, Pleidorina, and Volvox. But each one of these genera is polyphyletic. Uterina evolves repeatedly in this clade. Pleidorina evolves repeatedly in this clade. Volvox evolves repeatedly. So we're seeing a repeated evolution of genus-typical morphology and this repeated evolution of somatic differentiation. So a lot of my work focuses on a species of Uterina that falls right here in the tree, in this little Uterina subgroup that's an outgroup to the rest of the EVP clade. But I've also looked at other Uterina species distributed in different parts of the EVP clade. And during my PhD, I discovered that Uterina can develop plastic somatic cells in response to environmental stress. So I took Uterina and I subjected it to 2 hours of cold stress. And I found that some colonies will develop these small cells that are terminally differentiated. You can see them here. They don't reproduce, they're alive, they're indistinguishable from the somatic cells of other species. And this is a cell level response to the environment. This differentiation is under cell level control. Additionally, the offspring of some of these cold shock colonies also will have 1 or 2 somatic cells. So this is a group level response to an environmental stress. The development of these colonies is changing in response to the environmental conditions that their parent experienced. But when you look at the grand offspring of the cold shock colonies, they're indistinguishable from controls. So this is an example of developmental plasticity. It's being regulated by cell responses to the environment and organismal developmental changes. I told you that I looked at multiple different Uterina species. So I scored all Uterina species as either having facultative or plastic somatic cells, or as being uncertain if I didn't have any data on them. And I did ancestral state reconstructions to figure out if the ancestor of the Uterina clade likely had plastic somatic cells. And I found that the ancestor of this EVP clade could develop somatic cells, but it's unclear if they were likely to be obligate somatic cells or facultative somatic cells. I think because this is a false dichotomy. We know that some species that have obligate somatic cells develop more soma in response to environmental stress. But so it looks like there's changes in the regulation of somatic cells happening in this tree. But can somatic cell regulation actually evolve through changes in plasticity? Did genetic accommodation shape the evolution of cellular differentiation? So to test this, I've been using experimental evolution. Been focusing on a species of Uterina called NES 3984, whose genome is sequenced. I subjected to 2 hours of cold shock, which is my standard treatment that I stumbled upon during my PhD. And I cold shock them for 2 hours every 2 to 3 generations. That's every 4 to 5 days. And I repeated this between 5 and 15 times in separate experiments. I also assessed survival using the vital stain trypan blue. So here you can see a colony that's been stained with trypan blue. All the dead cells are purple, the living cells are green. I also assessed the phenotypes months after the last time they were culture shocked, and I subcloned 3 experimentally evolved lines. And I have thus far sequenced and analyzed the sequences of a control and an experimentally evolved subclone lineage. So I found that somatic cell development evolves really rapidly in response to repeated cold stress. So here you can see 2 lines that were repeatedly cold shocked. This is 5 months after the last cold treatment. And you can see there's a significant increase in how many colonies have at least one somatic cell relative to the controls here. Additionally, I subcloned these lines to see if there was genetic variation within them. And I found that there was. And that one of the lineages that I subcloned and then propagated was basically obligately differentiated. So more than 95% of the colonies in this lineage have somatic cells. And this is line 11A5, and it remains stable now years after the last time it was cold shocked. And you can see images of them here. Now, in a separate experiment, with a slightly modified number of rounds of selection, I found really the same thing. Somatic differentiation evolves really rapidly. There's a significant increase in the proportion of colonies of somatic cells, and I was able to identify subclone lineages that are consistently differentiated. I also found that the number of somatic cells per colony evolved. So here you can see the number of cells and the proportion of cells that are somatic cells for our controls. Controls are mostly 16 cells. There's sometimes 8 cells, occasionally 32 cells. They weren't here. And only a small number of cells are somatic. To compare this to experimentally evolved 11A5, you can see there's a huge change in this distribution. So what's happening is that the proportion of somatic cells per colony is increasing. So you're not just seeing increases in how many colonies have somatic cells. You're seeing increases in how many somatic cells those colonies have. Some of them have up to 80% somatic cells. And this is consistent across my subclones. So if you look in the controls, about 16% of cells in a differentiated colony are somatic. In the experimentally evolved lines, about a quarter of cells in a differentiated colony are somatic. And on average, in these obligately differentiated subclones, between a third and 40% of cells in a differentiated colony are somatic. And this is very consistent with the proportions that we see in the obligately differentiated Pleiodorina. This is really shocking, actually. Pleiodorina, on average, has between a 25 to 40% of cells in a given colony that are somatic, meaning that the primary morphological difference here lies in the overall number of cells they have. Pleiodorina has more cells than Euterina. So how repeatable is this? Was this just a fluke? Were these results really dependent on my selection regime? So I set up a cure at the University of Arizona last year. This is a course-based undergraduate research experience. So each semester, I have hundreds of introductory biology undergraduates experimentally evolving Euterina. And the first time they did this, which was a year ago, we did 3 different treatments to see if these results are robust to the specifics of the treatment conditions. So in treatment 1, colonies were cold shocked once a week, and this was repeated 5 times. So they experienced 5 rounds of selection. In treatment 2, they were cold shocked every 3 to 4 days. This is more consistent with the treatments that I have previously done. And this was done for 5 weeks, so 10 treatments. And in treatment 3, they were cold shocked again every 3 to 4 days for 3 weeks for a total of 5 treatments. Here you can see the pooled results. So in each one of these treatments, there are significant increases in the proportion of colonies with somatic cells. They are responding to selection really strongly. But the most effective treatment is this one with greater spacing between rounds of cold shock. So treatment 1 was significantly more differentiated than the other treatments. I think because there's more opportunity to respond to selection here. I'll get into that later. Then I looked to see if the number of somatic cells also evolved here. So here you can see the controls. On the x-axis, I've got the proportion of colonies with somatic cells. On the y-axis, we're looking at the mean proportion of somatic cells per differentiated colony. And there's no relationship between the two in controls. The other thing to note here is that each little circle here corresponds to data from one student group. So each dot is one lineage and about 50 colonies. In the experimentally evolved lines, though, there is a significant correlation between the proportion of colonies with somatic cells and the mean proportion of somatic cells per differentiated colony. But not all of these students groups successfully saw changes in the proportion of colonies of somatic cells. Not all of these student groups actually evolved changes in differentiation. So what happens if we separate out the student groups that have significant changes in how many colonies have somatic cells versus the groups that did not have changes in how many colonies have at least one somatic cell? So we see that the relationship between the proportion of colonies of soma and how many somatic cells those colonies have is driven by these groups that saw significant increases in the proportion of colonies of somatic cells. So here you've got data for teams that found changes in cellular differentiation. For teams that did not find that there were any changes in the proportion of colonies of somatic cells, there is also no changes in the mean number of colonies of somatic cells per colony. So there is this correlation between how likely a colony is to have somatic cells and how many somatic cells it develops. Is this because the underlying genetic architecture, or could this be driven by selection? Those are not mutually exclusive possibilities. So I looked at the fitness benefits of somatic cells. I assessed survival following cold shock in a species of Butyrrhina that's really closely related to the one that I did the experimental evolution with. And I found that more reproductive cells survived in colonies that developed at least one somatic cell. I also found that colonies that developed somatic cells had more living reproductive cells overall. So your reproductive cells are more likely to survive if some of your cells differentiate into soma. Then I looked to see if having more somatic cells is associated with greater reproductive cell survival. So here you can see on the x-axis, we've got the proportion of total cells that are soma. And on the y-axis, we have the proportion of reproductive cells that survive. So cells can be living soma, they can be dead soma, they can be living reproductive cells, or they can be dead reproductive cells. So I'm looking at how many living reproductive cells you have here and the proportion of your reproductive cells that are alive. And I found that having more somatic cells is associated with having significantly better survival of your reproductive cells. But this doesn't translate into higher fitness. There's important trade-offs here. So first, when you're developing somatic cells, you're automatically going to have fewer reproductive cells because cells can either be somatic or reproductive. So the more somatic cells you have, yes, your reproductive cells are more likely to survive, but you're going to have fewer of them. So it doesn't translate to overall significant fitness benefits. Additionally, the highest fitness is present in colonies that have no somatic cells. And colonies that have no somatic cells, they tend to either have the highest fitness or the lowest fitness. Now, this is a key trade-off, and really the only way to successfully circumvent this trade-off in a population of Euterina is to increase your overall cell number, basically to evolve something like a Pleaterina-like morphology. Now, I looked at survival following cold shock after multiple rounds of selection as well. And I found that repeated cold shock imposes very strong selection. After 3 rounds of cold shock, only about 80% of my colonies are surviving. There's massive death associated with this. And often, colonies that have at least one somatic cell are more likely to be surviving that cold shock than undifferentiated colonies. Okay. Next, I looked at the genomic changes that occur. I used long-read sequencing, and I examined my obligately differentiated line. This is line 11A5, and a corresponding control. I identified sample-specific mutated genes in both of these lines. Then I looked at the homologs of these genes. I looked to see whether the Volvox homologs have somatic-specific expression. Remember, Volvox is differentiated. And I looked to see whether the chlamydomonas homologs of these genes were expressed after cold shock. And finally, I compared the annotations of my genes to the known cold shock pathway for chlamydomonas and arabidopsis. So in my experimentally evolved line, I identified almost 4,000 unique fixed mutations. In my control line, I identified almost 6,000 unique fixed mutations. This is to be expected. This difference in number is to be expected because my experimentally evolved line was subcloned much more recently, and I wanted to get a full picture of the genetic variation that's present in the controls. So the controls are adapting to culture collection conditions. I found that the vast majority of these mutations are intergenic. And I identified 23 genes that have mutations in 11A5 and no mutations in the control, and 39 genes that have mutations in 1A and no mutations in 11A5. And I was able to annotate most of these genes. When I looked at the Volvox homologs, I found that in 11A5, the majority of these genes have a Volvox homolog that has somatic-specific expression. So we're seeing changes in genes that are expressed in the somatic cells of Volvox carteri. I also saw that in the control line, there are fewer genes that are somatic specific that are mutated. That difference is not significant. And I think what's happening here is that in the controls, we're selecting for motility because we're propagating the colonies that are able to swim near the top. So we're selecting for things that have better flagellar motility, and all flagellar genes have somatic specific expression. Next, I looked at the chlamydomonas homologs of these genes. And I found that in 11A5, almost 75% of the genes that are mutated only in the experimentally evolved lines have chlamydomonas homologs with expression in these cold shock pathways. So this cold shock pathway is being substantially modified And this is significantly higher than what we see in the control, where there are, significantly fewer genes associated with this cold shock pathway that are mutated. Then I mapped these annotations to the cold shock pathway. So this is where I really got a sense of what is changing in my experimentally evolved line. So up here, you can see the main steps of the cold stress response pathway in chlamydomonas. So first, I looked at changes in transduction by signaling molecules. So when chlamydomonas is cold shocked, a bunch of calcium floods into the cell, and that acts as a key second messenger mediating the cold shock response. And I found that there are changes in two cation channels that are probably transporting calcium. One of them is a voltage-gated cation channel with intron TE that has two known transcripts, and another is a potassium-dependent sodium-calcium exchanger. So there's changes in how calcium enters and leaves the cell. Additionally, all of the genes here that are in blue are genes where there's a Volvox-Carteri homolog with soma-specific expression, and all of the genes in italics are ones that have a chlamydomonas homolog that's expressed after cold shock. Next, I looked at genes that might be cold responsive regulatory factors, and I found that there is a serine threonine protein kinase with a promoter SNP. Then I looked at changes in transcriptional regulation, and I think this is where we're seeing really major modifications. And I really want to draw your attention to this gene here. This is 603. 603 is a squamosa binding protein. Squamosa binding proteins in plants are they're transcription factors that are developmental regulators that often mediate the transition between reproductive and vegetative states. This particular SBP gene also had a BTB/pause domain. These are repressors. So this is a transcription factor that most likely acts as a repressor. And in chlamydomonas, the homolog of this gene is downregulated in response to environmental stress. So in my uterina gene, there's a multi-exon deletion in a CDS SNP. The DNA binding domain of this transcription factor has been completely lost. So basically, you've got this transcription factor that's a developmental regulator that's normally downregulated in response to cold shock that is no longer present, which might be playing a role in, say, the constitutive activation of the cold shock pathway, potentially. There's also a nuclear importer with 17 SNPs here, and I think this represents secondary adaptation to developmental and genetic changes. Then there's a lot of changes in genes that are associated with coding for protective proteins and degrading mechanisms. We've got a universal stress response protein with an intron TE, an oxidoreductase with a transposable element in that promoter. This is potentially quite significant because we know that redox reactions are likely to play a role in cellular differentiation in Volvox-Carteri. There's also changes in a heat shock protein in ferredoxin domain containing proteins, indicating some potential involvement in photosynthesis. We've got an FBOX protein, so that might be representing changes in protein ubiquitation and degradation. And we've got the sulfite exporter with a SV insertion in the UTR region. There's also two more clusters of genes that have been modified that aren't part of this canonical stress response pathway. So we see a set of genes that are involved in cell cycle regulation that are evolving. And a lot of these are involved in apoptosis and programmed cell death, which is significant because the somatic cells in differentiated bulbocene algae species, well, they die as a key part of their life cycle. The reproductive cells hatch out, the somatic cells undergo what we think might be programmed cell death, right after those reproductive cells hatch. There's also changes in cell wall remodeling, which is potentially significant because cell membranes and cell wall annotated genes are significantly enriched in cell type specific expression in Volvox carteri and in another species that has independently evolved differentiation, which is Astrophobony. All right, so that was a lot. Let's bring this together. So what did we learn about how the modification of plastic responses might have shaped the evolution of differentiated multicellularity? So I showed you that multiple Eutorina species can develop these plastic somatic cells in response to environmental stress, and that the capacity to develop somatic cells is likely ancestral to the EVP clade. I showed you that repeated cold stress results in the rapid evolution of obligate somatic cells through the stabilization of the plastic response, and that the proportion of somatic cells per colony also evolves really rapidly. So while I conceived of this evolutionary refinement step as occurring after the step of the loss of plasticity, they might be occurring, looks like they are occurring simultaneously. And these changes are driven by selection that's favoring somatic cell development. And in at least one line, this is occurring via the genetic modification of the cold shock response pathway, as well as changes to how cells progress through the cell cycle. This allow us to think about how development evolves. So let's think about development in standard Eutorina culture collection conditions. So we're going to start with a single here in a colony. It's going to divide, undergo inversion, become larger, and then hatch out to form the next generation and grow and so on. We can conceptualize this through this simple pathway where development starts from a single cell and it typically proceeds along a pathway resulting in an undifferentiated organism. But about 10 to 20% of the time in normal collection conditions, development proceeds along this pathway that results in somatic cells being present. We can further conceptualize this through as a ball rolling down a hill. This is Waddington's epigenetic landscape. And the shape of this landscape is just conceptual here. Now, cold shock changes the shape of that landscape such that now development is basically equally likely to proceed along either one of these two pathways. When I experimentally evolve my Eutorinas, I find that in these populations, development consistently proceeds along either of these pathways without the need for that external environmental signal to modify development. So the changes in developmental landscape that were previously induced by cold shock are now stable. In my experimentally evolved line that has obligate differentiation, development consistently proceeds along this pathway that results in somatic differentiation. And the pathway that leads to an undifferentiated state is much less accessible. Similar to what we see in Pleiadorina, where development consistently proceeds along this pathway, resulting in differentiation. And although I don't show it here, sometimes you do find Eutorina that have no somatic cells. So somatic cell differentiation can really rapidly and repeatedly evolve through the modification of developmental plasticity. And I've identified a new pathway through which somatic cells can develop. We know a little bit about the genes involved or the genes necessary for somatic cell development in Volvox carteri. And those genes were not mutated in my experimentally evolved lines. That's not to say they couldn't be differentially regulated downstream, but there's no genetic changes in those. So we've got this novel pathway resulting in the evolution of somatic differentiation. And I also found that changes in the development of somatic cells co-occurs with changes in the proportion of somatic cells per colony. So I think that in the EVP clade, which is also called the uterina clade, the convergent evolution that we see, the repeated evolution of uterina, claderina, and volvox, might be underpinned by the repeated modification of inherently flexible developmental landscapes. Uterina can develop to be differentiated or undifferentiated. And it's really easy for selection to act on differentiation and result in an increase in the proportion of somatic cells such that your proportion is similar to that of Platerina. Platerina has a wide array of morphologies that it can develop in response to different environments. It can increase the proportion of somatic cells in response to things like nutrient deprivation. We also see Platerinas that have a really small proportion of somatic cells. We see some Platerinas that have a proportion of somatic cells that's very similar to that of some Volvox species, like Volvox powersii. Sometimes some of these Volvox species also have a smaller number of somatic cells than you would expect. So you can see these transitions between these two genus typical states. And I think these are happening by the modification of which developmental pathways are predominantly followed, at least as far as the proportion of somatic cells goes. Then in some lineages, we see the evolution of this derived developmental program called asymmetric division. This is what Volvox carteri has. And in the EVP clade, when asymmetric division evolves, you never see the secondary evolution of, say, Platerina-like morphology. You get terminal differentiation. The only way you see undifferentiated looking, or the only way you see Volvox carteri that has a smaller proportion of somatic cells is really when they have a mutation that causes the loss of obligate differentiation. So this derived developmental program is really entrenching or canalizing developments. So I've got a fair bit of ongoing work. So first, I have ongoing genomic and image analyses. I have 2 more sequenced genomes that I haven't yet analyzed, and more image analysis to better understand how selection shapes the repeated evolution of differentiation. I also have 500 to 800 students at the University of Arizona that are experimentally evolving uterina each semester and collecting data to address a new research question. So last semester, they collected data on reaction norm evolution. So I'll be able to look that I've got all the images for that. So I'll be able to look at how the plastic response to the environment evolves in response to repeated cold shock. And right now, they are using our most effective selection regime. And they're going to send me the samples, and we're going to look for convergent genomic changes. And more broadly, I'm really interested in understanding how genotype-phenotype maps evolve during the evolution of obligate somatic differentiation. And how do fitness landscapes shape this transition? How do the really variable developmental and phenotypic landscapes that we see and an array of different species shape this transition. And how do these processes interact with each other? So I have a lot of people to thank who've been involved in this work. I started this work during my PhD at the University of Arizona. So I'd like to thank my PhD advisor, Rick Michaud, number of folks in the Olson Lab, first at Kansas State University, and now at Augusta University, were involved in the image analysis or in the transferring of the algae. And then primary person that I want to thank is my collaborator at the University of Arizona, Ryan Rubiones, who runs the introductory biology program, where they've been experimentally evolving these lines, and all of the introductory biology students who are doing this experimental evolution work. And I would like to thank funding, especially the National Science Foundation, who funded my PRFB. And I'd like to thank all of you for being here. And I'll take any questions you might have.